Hello, everyone, and welcome. I see so many of you already jumping in on the chat, and it's so great to see you. As always, a big hearty welcome on behalf of the Consciousness and Healing Initiative. My name is Dr. Shamini Jan. I have the pleasure of being the founder and president of this beautiful collaborative of scientists, healing practitioners, educators, artists, and innovators who lead humanity to heal ourselves through the exploration of the biofield in very large part. And we're so excited for today's guest. As you pop into the chat, just so our guest has a sense of where the community is hailing from, please feel free to jump into the chat as you typically do and introduce yourself and where you're coming from, just so we know. We've got Julie here from Florida jumping in. Hello, Julie. Nice to see you. Pakistan, wonderful. New York, Ohio, Lincoln, um, Ventura, Florida, Wisconsin, San Diego, Minneapolis. Fredericksburg, Scranton, lots of US folks today. This is wonderful. Danbury, Arizona. So beautiful to see all of you, and those of you joining us internationally as well, welcome. Auckland, New Zealand. Yes, we were just commenting that it's probably quite late for you there. Thank you for joining us live. Wonderful. Canada. Hello to our beautiful Canadian neighbors. And France. Wonderful. Serbia. So glad that you can all join us today for such an incredibly, what's going to be such an incredibly powerful presentation and dialogue on cutting edge science in the biofield and what it might mean for us as we consider how powerful our consciousness is in today's world. I have the pleasure of introducing a dear friend and colleague who also serves on the Scientific Advisory Council for the Consciousness and Healing Initiative. His name is Dr. Arnaud Delorme. Arnaud has been studying human consciousness for the past 2 decades, probably many lifetimes, is my guess. He's a neuroscientist at the Institute of Noetic Sciences, a CNRS research director in France, and a senior research scientist at the University of California of San Diego. In 2000, Arnaud completed his PhD thesis on visual categorization in humans, monkeys, and machines. And a lot that I could say about him. He did his postdoc at the Salk Institute in Terry Sevnovsky's and Francis Crick's laboratory, where he focused on statistical analysis. This is very relevant to our conversation today, of electroencephalograph signals during various cognitive tasks. He actually developed the free EEG lab software. For those of you who know psychophysiology, you know that this is one of the most widely used open source softwares. Arnaud chose to make this open source, and it is a software for the advanced analysis of EEG signals. This was in collaboration with his colleague Scott McCaig. He has been awarded many awards, including a Breton-Schuler Young Investigator Award, a 10-year anniversary ANT Young Investigator Award. He has been at the forefront of research in consciousness and spirituality for some time. Arnaud comes to this both as a scientist and a longtime Zen meditator. He is the author of over 160 publications, including work on mediumship. And today, we're super excited to talk about some of the most groundbreaking work that is happening in the field of biofield science and healing, where we are exploring the biofield and electrophysiological connection between healers and their targets, which are sometimes cells, as we'll learn more about in this study. So, Arnaud, thank you so much, first of all, for being here. Really grateful for you to take the time. I know how busy your schedule is and with all of the incredible things you do in the science world. So thank you.
Thank you for having me.
Yeah, really just so delighted. I literally jumped out of my skin with this latest publication. I tend to get very excited about your work anyway. And I'm really excited to dig into this work today and, maybe talk about how it's feeding into a whole program of work, desired program and building off of things that we have done in the past in the biofield science world and taking it further, understanding how it might relate to the threads of consciousness that connect us, from humans to cells, and even implications for technology. There's so many places that we might go in your presentation and our discussion today. I do want to remind people that you have this incredible opportunity, those of you who are joining live, If you have questions for Arnaud, we will get to them toward the end of the hour. If you're joining us here on Zoom, you just go to the bottom of your screen and you'll see a little box that says Q&A. Q&A. You'll click that box and type in your question as we go through Arnaud's presentation today. Please don't put it in the chat, because if you put it in the chat, we will probably not be able to see it. So do enter it in the Q&A. But we'll start with a very simple question for you, Arnaud, as you launch into your presentation, which is, as a Zen meditator, what drew you to the biofield and your interest in this particular study even?
Well, that's a deep question. And I think, as you already mentioned, it's linked to the study of consciousness, what consciousness can do, what it cannot do. And, the scientific study of consciousness, that's what drew me to, work on this topic.
Through
And
the exploration of your own, and as you know, many of us in the scientific field will question what this relationship is with consciousness and energy. I mean, Arnaud, I know that you know the Mind and Life Institute very well, and gee, I don't know, this was maybe at least 15 years ago, maybe more now, who knows, that I went to a summer institute at the Mind and Life Institute. And, the usual players were there. When we brought up these questions about extended consciousness and energy, we were sort of woven away, and there's this sense of, we don't talk about that. We don't talk about extended consciousness. We don't talk about things like the biofield, always this waving away, this fear of talking about something that could be viewed as pseudoscience. And yet your data that you're presenting today is so robust. It's been published in one of the top journals in the country, a nature journal, Nature Communications. Is that right?
a scientific report, yeah, which is in.
Scientific reports. So maybe we've come a long way, baby. But in your view, how would you, if someone asked you, what's this relationship between consciousness and energy? And how might it relate to the kind of research that you do? What would you say to that?
um, well, I would say that's the crux, of studying consciousness. How does consciousness interact with the world? Through healing, through other studies we do at IONS, telekinesis, telepathy. Telepathy is really hot topic right now with the telepathy tapes. Yeah, that's the interface where, that needs to be studied. Exactly, consciousness and, what you call energy, whether it's, the energy of healing or, the energy of physics, just like the definition of energy. So yeah, I think it should be more at the center of consciousness research than it is right now, because right now a lot of consciousness research is just debates which have no outcome. So we're trying to bring like experimental view, like experimental in experiments, trying to do specific experiments. Well, can consciousness do that or not? And yeah, trying to answer these questions.
Yeah, so actually really looking at the application in the real world. And as you know, as a clinician, I'm deeply interested in knowing, how do we direct our consciousness in a way that can facilitate healing for others? And how does that work? I mean, that's just the golden question, right? How does consciousness interact with the world and affect physiology, affect cell signaling? And so we're so excited for you to walk us through some of this latest data that you have published in collaboration, I believe, with Dr. Lorenzo Cohen, who we have had on Chi webinars before, and of course, as part of the Science of Healing course, where he has shared a bit about some of the research that has been coming out of MD Anderson over the past couple of years. And I understand this was in some collaboration with that. Just to give people some context, and we can send you resources or check out the Science of Healing course if you want to learn more and hear from Lorenzo on this work himself. But this builds on some research with the same healer and other healers, as I understand it now, where in these carefully controlled studies, Lorenzo's team was able to demonstrate that this same approach, this same biofield approach, was also showed some strong evidence for reducing the spread of tumors in the specific mouse model of cancer, right? So those studies are also published in scientific reports. And you're not going to necessarily talk about the effects on cancer metastasis today. So that is a different set of papers. So just kind of letting everyone know that while these threads are tied together, today we're going to focus very specifically on the papers and the studies that you led looking at the electrophysiological connections between healers and cells, correct?
That's right. Yeah.
Great. Well, please, yeah, take it away. We're so excited for your presentation. Kind of walk us through what you've done and what we're learning about the field and our field connection.
Okay. So yeah, let's get started. First you were saying, oh, what brought me to study these questions? And so I don't want to go back to the Big Bang, but when I grew up in France in the Paris suburbs, and I think I was in seventh grade, and I got this in the courtyard at my school, and I was like, I didn't know what I wanted to do, professionally. At that age, you don't really ask yourself this question, but the thought struck me, oh, I'm standing here, um, who I am, was the main question. And in my upbringing, if you wanted to answer these questions, well, you would need to become a scientist and study the brain. And so that's, maybe if you were born in India and you become a monk, but in the Western world, you just try to answer the scientific way. So that's what I did, and I became a brain scientist. And then what I'm most interested in is what we call big C consciousness. So there's little c consciousness. That's ChatGPT. That's the thing, that just can work on computer. That's parsing images, understanding sentences. And there's the big C consciousness. The big C consciousness is the consciousness of what we call qualia, the sense of seeing, of being, and that's the real mystery. And actually, there was a paper in Nature, I think 2 years ago, big C consciousness. The problem of understanding consciousness is as big as a problem as dark matter, for example. What is dark matter? So this was ranked along the same lines. So even in mainstream physics, there is a lot of interest for that specific question. And we also, sometimes it's named the hard problem of consciousness. There's the easy problem of consciousness. That's ChatGPT. Again, it's like how can, it's like neurons solve computational problems. And I think, the latest development in AI show us, well, artificial neurons can do it, biological neurons can do it, but the hard problem of consciousness is this sense of existing. So, I like this diagram where we ask ourselves some questions, but let's define our theoretical framework. Where are we? So you can say you equal your brain, and if you answer yes, you're what we call a materialist reductionist. If you answer no, uh, then again, there's two possibilities. So, there's the neural responses right here. And their neural responses, the standard view is that, they gave rise to consciousness. And consciousness is an epiphenomenon. It just emerged from the interaction of neurons. Uh, and if you ask some mainstream neuroscientists, they'll say, well, we don't know how, that just happens. And again, there's two view there. Either it emerge and then it can have, so consciousness can have an influence on the neural response, or it just emerge, it's just epiphenomenon, so it can't have any uh, uh, it can't have any causal influence on the neural response. And if consciousness is epiphenomenon, it cannot have a role on the neural structure, then there is the absence of free will. And, just to give you an idea, I would say materialist reductionist, you have like, this is where most scientists were in the 1980s. Now, I think this is a smaller camp, it's 20% of scientists, but, 70% are in this camp, absence of free will. And yeah, it makes no sense from a felt perspective. We feel, we have free will, but that's where most neuroscientists and scientists lie. Uh, um, and then there's the alternative hypothesis. We are more than our brain, and our consciousness has causal influence in the world. And I think that would be the remaining 10%. So this is where I put myself. I think Chamonix puts herself as like consciousness can have a causal influence in the world, and that's how you can have healing. And again, if you're in that camp, you still have one last branch in which, okay, we can be more on our brain, and our consciousness can have a causal influence, but maybe it just interacts inside the brain or something. Or it can have a non-local influence outside the brain. And so that's psi healing. And so this is, where we are. Or if you answer no to that question, it just happens with the brain, and you have different models, which are consistent with this hypothesis. And, it sounds I don't know how many of you have been involved with the Chi Initiative or IONS, but, it sounds crazy. Psi, which is basically telepathy and healing, how can that be real? Well, there is other crazy theories which have been demonstrated, or at least are widely accepted. For example, non-locality is everywhere. You can have entangled atoms at the opposite side of the universe, and they would interact instantaneously. So that's called entanglement, faster than the speed of light. So there's really weird stuff happening. Black holes, like time collapse. So just to give you perspective, it might, if you're coming from mainstream, there's like, oh, it's impossible. There's a lot of weird stuff in science already. And, we can't claim that we understand it all. And this is here what we call our atom strategy. So atom strategy is we're trying to maximize, the discovery of this effect, psi and healing. For this, we try, we can try different things. We can try to change the analysis. We can try to change the target. We can try to change the operator and the modulator. So, for example, the target, you can use your mind to try to influence quantum system, or water, or stem cells, or DNA. So in this case, it will be specifically other humans and cells as well. And then we can try to change the analysis. So here we're doing like a standard analysis. And the operator also, who is trying to do that also matters. So here we'll test a specific talent, as Chamonix mentioned, and I'll give more details about that. And here we're not dealing with, trying to modulate that. Like, for example, we could give psychedelics to the talent, and maybe it would do better. But we're not trying that here. So these are how we're trying to maximize that effect. And first, because before I delve into that, I want to show some results which are now, widely accepted, that we interact with our body interact with our environment. This was published just a couple of years ago, 2021, and it's the fact that women menstrual cycle align to some extent with moon phases. And if it sounds obvious, it's about the same, similar time frame, but I don't think it was developed, it was demonstrated scientifically before that. And that's a direct influence of the planet on our body. Here is another one where they put a subject inside a cage with all these wires to simulate magnetic fields. We knew pigeons could orient themselves with the magnetic field, so they have magnetoreceptor inside their head so they can sense the Earth magnetic field. And so that's how they orient themselves and can fly thousands of miles um, across the planet. But here, this study shows that humans also have magnetosensors, so they can also sense the Earth magnetic field to a small extent. And then this other study by our dear collaborator at ArtMath show that HRV, so heart rate variability, changes based on sunspots and solar wind. And again, these are very robust results published in reputable paper. And I don't think they're that controversial anymore, especially if you imagine, for example, you have solar wind, which is going to affect the Earth magnetic field. And we saw that we're sensitive to magnetic field. So there is even a mechanism through which this could happen. And also, again, this shows, we are connected very strongly with our environment. This is a survey here we published on meditators. So we asked people to meditate, and here you could see, I feel more than half of the people have felt a connection with a teacher, of a guru, of a supernatural nature, or clairvoyance and telepathy. So this is not something rare. I mean, it doesn't mean their experience is real. It's just something they felt, but it's not something rare. So now show me the data. And so this is the paper that Chamonix was talking about. So this is the first paper we published with Lorenzo Cohen at the MD Cancer Center in Texas. So at the MD Anderson Cancer Center in Texas, actually the first premier, I think it's #1 in the country as a cancer center. So it's a very reputable place, and it's incredible they were able to run the study over here because they acquired the data, and basically I processed the data and analyzed it. And this was financed by the Emerald Gate Foundation. So Biofield therapy. So here my expertise is more in EEG processing, and I just arrived in biofield therapy. Chamonix has been one of the person who's published the most on that, so I'm very honored to be here. And there's also been some publication before on cancer. So I just wanted to give you there's a lot to read, and Chamonix can put the link to all these papers. So there's prior work. It's not we're starting from nothing. And I think that's also, as a researcher, I remember when I started being interested in these topics, you're very arrogant. You think, oh, nobody has ever done that. But there's tons of study that are very serious, and it's worth to go through the literature to see what else is there. So our study is just one of many. And so this is the first study. I'm going to show you two studies. This one was with cells, and the other one's with mice. So this is John Lovac here, who was trained in the Bengtson rapid cycling method. And you see here is trying to heal, so without touching, is trying to heal two sets of cells. Some are under this microscope that measures different measures, so 3 or 4, and calcium or beta-actin. And this is another incubator where there are more cells. So these are cancer cells, and he's trying to heal the cells. And there's also control cells, control petri dish. And he doesn't know. He doesn't know what he's trying to do. But the idea is that we want to show there is a difference when he's trying to heal the cancer cell versus the non-cancer cell. The technique he's using is actually interesting. So it's called rapid cycling. And I was talking to Dean Radin last week because Dean trained on this method. Also, we're sad to say that Bill Bengtson actually passed last month, so the inventor of the method. So this is John Lovac, is a star student. And so the way this method works at least as described by Dean, is you're wanting you want to so the you imagine things that you want. So for example, you want a Lamborghini, and you're going to visualize the Lamborghini. You have a picture of Lamborghini, and then you do this with many things you want. For example, you want a great meal, and then you're going to visualize the meal, or you want to be rich or something. So it's just very egotistical. And then you do rapid cycling, in which you work with the image and you focus on the things you want and you cycle through them rapidly. And the idea is that as you cycle through them, only the want stays. And basically that carries, the healing intention is the want, because as you're cycling through them, uh, you can't remember exactly all the details, and you just focus on the want, and that's what helps heal the cell. Again, I'm just, repeating what Dean told me. Maybe some of you have been trained in technique, and be happy to hear what you have to say about it. I wasn't trained in it, so. Uh, so this is what he's doing with the cell. And this is how we design uh, the experiment. So what we do is that we have a baseline at the beginning and a baseline at the end, in which the cells are not even there. And so we are measuring the brain waves. You can see here is hooked up with this system that's measuring his brain waves. And we're also measuring the cell activity with these microscope measurements. So we have a baseline measures. We have 3 periods of treatment, so it lasts for 15 minutes. And the reason is we have three periods is so it can rest a little bit in the middle. And then we have a post baseline. And these are the cell measures we're measuring during the treatment. And then we do the same with control. So these cells have cancer, and these cells do not have cancer. And again, we do pre and post baseline. So we have basically 4 conditions. We have baseline with the cell that have cancer. We have treatment when the cell have cancer. We have baseline with the cell which don't have cancer, or controls, and we have treatment with the control. So it's what we call a 2-by-2 design. Two baseline treatments, that's one condition, and cell with cancer, no cell with cancer, the other condition. And that's what's represented here. You see baseline treatment. So, here we group the 2 baseline together, and we group all the treatment together. And then control cells and live cells, these are cells which have cancer. And these are scalp topographies in the theta frequency bands. So here this compares baseline versus treatment. And basically we want this not to be green. When it's green, is nothing is what we call significant. And significant means statistically significant, which is what we're interested as scientists. As scientists, we try to show, okay, there is one chance in 1 million that this is due to chance. It's just a random fluctuation in the data. So when it shows some color here, it shows like, okay, this is like some kind of real effect. We didn't find, so we find baseline versus treatment when we group live and control cells. We didn't find a difference between live cells and control cells. Here it says NS means not significant. And we did this in theta, and we did this at other frequency bands. Now, what's interesting, so for all frequency bands, we found difference in the brain of the healer when he's healing versus not healing. So we find a strong difference here in theta, alpha, beta, and gamma. But we only found a difference when he's healing live cells versus control cells. Here, with single electrodes in the beta band, so beta brain waves are about 15 hertz, and in the gamma, which is even higher. So we found this peripheral, which is not necessarily convincing, even though it's significant. It's just one electrode. It would be nicer. It's like this. You see here, everything is significant, but still, there is some difference when he's healing control versus live cells.
So just to unpack that a little bit, make sure that everyone is following, because this is beautiful and lots of really great graphs here. When we're looking at these heads, On the right, yeah, and I'm using my pointer, but I'm imagining people can't see Arna, but when you're looking at the baseline versus treatment p-values and you see all of this color here, this is basically showing that if the healer is going into healing mode, the brain, there's some market difference, it seems, between the baseline versus treatment, regardless of what kinds of cells he's working with, whether they're the controls or the live.
Yeah.
But in addition to that, there is a significant, statistically significant, but not all over the brain,
Yeah.
difference, which is this bottom, the very bottom graphs that you're showing us here, the green with a little bit of a red on the right here.
Yeah.
That's where you're showing the difference between the live and control cells baseline versus treatment. So this is showing the very, very unique effect of the healing on the actual cells with cancer, that it is actually different than the healing trajectory for the non-cancer cells, correct?
Exactly.
Hopefully I made that clearer. I don't know if I did, but then we're talking about statistical significance, and there's a lot that we could even talk about in terms of your methodology and how it's improving the field,
Yeah.
having these kinds of 2x2 designs, which has not always been done, but I will stop here. Please continue.
Yeah. And ideally, what we would also have observed, which we didn't observe here, unfortunately, is that ideally you want an effect that specifically when the healer is treating the cancer cell. So because here, when we say, oh, this is significant, it's just live versus control. There's no specific difference between baseline and treatment. So we run also this analysis. Okay, is this effect only visible for treatment, or is it also in baseline? It seems it's both. So, it's not ideal, but it's still something.
And
Um,
I mean, from a practitioner point of view, you could say we're looking at this, and the brain changes when you run energy.
yeah,
When you don't know what cells you're working with and you're running energy and intention to heal, the brain changes regardless of what the medium is that you're
exactly.
applying it to.
Yeah. Um, this one is also extremely interesting. Now we try to use some other statistical tool. It's like, is there like some transfer of information between the brain of the biofield therapist and the cell, and vice versa? And for this, we actually use a method that's called Granger causality. It comes from financial markets, where people are trying to predict, future financial market, or does the stock of Google influence the stock of Apple, or vice versa, etc. So this comes from this field, finances, and we can use it in brain analysis too, where we see, okay, do cell influence EEG, and do EEG influence cell? And so these are very robust methods. So here we see we had two cell measured, tubulin and calcium concentration, which are some measures of health. And we interpret, so this is more of a Lorenzo's, our collaborator as the MD Anderson Cancer Center expertise. I'm more the EEG person, but it does show some difference. Here you can see EEG influence cells for the calcium concentration over these electrodes specifically, uh, in the theta, alpha, beta, gamma, basically all the range, but not vice versa. And here for the tubulin, it seems that the cell influence the EEG in the beta and gamma range. So again, these are results on one patient, but this is promising, and we think this warrants trying to be replicated by other lab to see if.
This is huge. So, do you mind if we unpack this a little bit more to just to make sure that we understand it? Um, because you're saying influence. So you're speaking of causality now. You're not just speaking about these things being correlated. You're saying you're using a specific analysis, which has been used in financial markets to see what predicts what. So in this model of the Granger analysis, you're actually taking a time point. Let's say, for example, if we're looking here at tubulin in the cells, you cell to the EEG. You're taking a time point of the activity of the cell and you're inferring whether that activity in time is actually influencing potentially the EEG of the practitioner. That there is a causal, you're suggesting that there is some kind of causal relationship between the activity of the cell and the activity of the brain of the healer, that there is some causal communication. That's what you're suggesting.
Exactly. I mean, causal influence. Yeah,
Uh-huh.
because
And also, if we look, for example, on the calcium flux side of things, interestingly, you're not seeing the cell to EEG signal. You're seeing an EEG, meaning the healer's brain, right? So we're measuring the healer's brain with the EEG. And your Granger analysis is suggesting that there is a causal link between the brain waves of the healer, in this case, and the calcium flow of the cells.
exactly.
Yeah, that's pretty incredible. And for most scientists, it would be very hard to believe. So you mentioned that you think this needs to be replicated, right?
It always needs to be replicated. A paper by itself, means little. So that's the process of science is, replication. Yeah, I'd like to see it, replicated. And we published all the script analysis, we published all the data. So, people can exactly see what we did. So this was the first study. And now moving to second study. Same biofield therapist, John Lovac, right here. Same setup where we record his brain, but now he's trying to heal some cells, some mice. So he has 4 mice in front of him. Again, he's not yet touching. And here we're recording his EEG and the brainwave of 4 mice simultaneously, um, which is actually an incredible technical feat. I think it's the first, and we're measuring also this down to millisecond precision because we have a marking system where we send events so we can synchronize the data down to 1,000th of a second, which is required, by the way, to do the analysis I'm going to show you. But also, this is the first, to my knowledge, the first human, non-human recording of simultaneous data. Never been done before.
Wow,
And so this we're in the process of publishing, but we already have actually we're finalizing the paper. I
exciting.
finalized the last figure yesterday, so I
Oh,
can.
so you guys are hearing it here first. And this is very rare, by the way, that we've built enough trust with these scientists at this point that they're willing to share this data before it's published. That usually never happens.
Yeah, well, it's gonna be hard for people to reproduce this kind of setup. So, it's not like somebody's gonna, oh yeah, I see what you did, and then I'm trying to beat you by publishing first, which is like the
Wouldn't
game.
that be lovely? Like, look at 100 labs doing the same thing, and that's our dream.
Yeah, and also in our field, I think there's more collaboration. That's like it's not cutthroat science. I mean, we're trying to show, I'm happy if another lab show me before me and as it gets published, because it just goes in the direction we want it to go.
So the
So
first study to look at simultaneous EEG between healer and mouse.
heater and mice, but also human and animal ever. Um, so this is our setup. So we have a baseline where the mice are not even there. Then we have another baseline where the mice are there, but he's not treating them yet. Then we have a treatment period of about 30 minutes, and then we have a post baseline where he stops treating the mice. And we basically have 4 conditions. Um, we have the biofield therapist, so the one I showed you in picture, that's healing cancer mice. We have the biofield therapist that's healing non-cancer mice, and he doesn't know. Like in the first study, he didn't know what kind of cells were treated. So that's why it's called a blind design. And in addition, the person who brings the mice or who brought the cell in the previous study doesn't know either. So that's the experimenter. And that's what's called double blind. And that's very important to do double blind because if the experimenter knows his body language, like, maybe the way he puts the mice, like, oh, this is not going to work anyway, because I know these are not cancer mice. We're humans are we can pick up on these things. So that's very important to run it double blind. So both studies were running double blind and also blinded as the person who analyzed, they only told me at the end who was the therapist and who was the sham. So sham is non-therapist, and which were the cancer mice. So I did all the analysis, and I didn't know the outcome. I said, "Oh, I found something significant." I was like, "Oh, these are the cancer mice." So I was unblinded at the very end. And again, it's a big deal because it's much harder to analyze data this way. It's also very important to be very rigorous in this kind of
And
approach.
Kay,
Yeah.
and Kay is making the joke, talk about four blind mice.
Yeah.
But in reality, not to take us too far, but this relates to consciousness, of course, and what these mice are experiencing. W- Interestingly, this is not triple blind. We couldn't determine whether the mice knew. You would say, did the mice know whether they were getting biofield therapy or sham therapy? And I've reported on this before with Lorenzo's previous data that actually it does seem like they can tell the difference based on their behavior, and that was published. So you cannot do a triple blind study because guess what, guys? The mice can tell whether they're getting healing or not. And previous published papers from Lorenzo's group have actually shown that previously, when Lorenzo did a similar study with another healer, Sean Harabance, the mice would actually crowd to the front of the cage when Sean would come, and they would not do that for the sham therapist. It was like they could really feel and sense what was happening. So anyway, just a little aside there as we think about these experiments. And whether these mice, how conscious these mice are. Perhaps they're more conscious than we are. Anyway, sorry, go ahead.
And so the sham therapist is someone that's not a healer and that just, use their hand as if they were healing, basically. So we have some kind of baseline. And so this is actually the same analysis I showed you. So now he's healing, not healing the mice, not the cells. But we can do exactly the same analysis on his brainwaves. And the good news is that we did reproduce at the very least, baseline versus treatment. So we find very similar results. Not exactly the same, it's a different experiment, but in the all frequency band, baseline versus treatment. So that's the healer healing, trying to heal the mice. What's the difference in his brainwave when he's healing versus not healing? So this was reassuring. We didn't find any difference between the cancer and the control mice in this type of analysis. Meaning his brain waves, the brainwave of the healer, we're not looking at the brainwave of the mice yet. The brainwave of the healer is looking the same whether the mice have cancer or the mice don't have cancer. Um, and for control, here we see the sham healer doesn't have as much difference, but also has some difference, and that's understandable. They go to probably a different state of mind, very focusing on the mice compared to just resting. So we can expect also some difference, but the difference is much stronger for the biofield therapist than the healer. And again, these are qualitative analysis. So we can't compare. We would need a population of healer and a population of sham to really do some statistical analysis. So this is just qualitative. And now the interesting part is we looked at the brain synchronization between human and mouse. So for human, we have all these channels right here on the scalp, and for mouse, we have only 4 channels. And this is a muscle channel, so it's actually a neck muscle. This is measuring neck muscle. And so this is what we found, and I'm going to unpack this plot, which is relatively complex, where here we look at the correlation of amplitude. So whenever whenever we have an increase, for example, in theta, like 5 hertz, in the brainwave of the human subjects, do we have the same increase in theta in the mouse brain? So this is what this shows. So this is theta. So this is the first frequency band. In the first frequency bands, we don't observe any co-modulation of power. So this is co-modulation of power. When theta increase in one electrode, does it increase in one electrode in the brain of the mice? And to assess significance, if it's significant or not, we just apply some, powerful statistical analysis. And here we have treatment versus baseline. So here it shows no difference, no pairwise commodulation of brainwave amplitude in the theta band, treatment versus baseline. But we do have some for alpha, beta, and gamma. And we have some mostly with the electrodes here, that's neck electrode. And so what this shows is that there seems to be difference baseline versus, because the neck electrode is going to be very sensitive to the movement of the mice. And so it's very likely that when the treatment is happening, the mice behavior changes. And this is picked up by this electrode right here, which is a muscle electrode. Mouse type, this is cancer mice versus non-cancer mice. And again, we found a couple of significant changes in the beta bands. And this is here the column that's the most interesting, because this is what we call the interaction column. And the interaction column is when we want to know, well, is there a specific difference? Healer, biofield therapist treating cancer mice during the treatment period compared to everything else, when he's not treating them in the baseline or when these are not cancer mice. And so this shows some very strong, significant results here. And this is, what we call a marginal analysis. So this is the last analysis I was running yesterday to really show how it differs. So you see here, we have actually a stronger effect during the treatment than the baseline. So this is treatment, this column versus baseline. This is cancer versus non-cancer. And whenever you have a line, it shows like a significance. So everything is significant here. Control baseline is differently significant from baseline cancer. Treatment cancer is different from treatment control. So because we have lines here, everything is significant at the threshold we use. So this is the first analysis we did. And you see here, it's not only with the neck muscle, but it's also with one of the brain electrode right here and the alpha and beta band. And then this is, I think, the last plot I'll show. So the previous one was co-modulation of power. So it just means brainwave increase in the human. Does it increase at the same time in the mice? And this one is slightly different because this one is brain synchronization. So if you have your brain waves, they're not necessarily synchronized. They can be shifted, for example. This will test specifically if they're synchronized or if there is like a constant lag between the brain waves. This analysis doesn't do that. This analysis just shows our windows of interest, which I think is 1 minute. If you have increase in theta in the human, do you see also increase in theta in the mice? But this shows pure synchronization. So this is why we need the 1,000th of a second synchronization. So we can run this kind of analysis.
So this is literally whether the waveforms are moving together.
Exactly, whether they're moving together or not. So whether the brain are synchronized. And so what we see here, again, a strong effect treatment versus baseline with the neck electrode, but also with lots of the brain electrodes. So again, something happens in the brain of the mice when the healer is treating them versus not treating them. Although we didn't see specific changes, I don't have the slide here, but we also observed that in the sham. So we can't say that's specific to the healer.
Okay, wait, I just want to make sure we understand this. So if I'm following you, Arnaud, then what you were saying is when the mice, whether they have cancer or not, are getting treated by a practitioner, whether they're an actual biofield therapist or the sham therapist,
Yeah.
you see increased synchronization between the healer, whether it's a sham healer or a regular healer, and the mouse. So when there's this intention, ostensibly, we didn't talk much about what the sham practitioner is doing, but let's just assume that the sham practitioner is actually trying to intend to heal the mouse. Then that's when you see a synchronization of the brain between the mouse and the person. Is that right?
Yeah, that's right. So here, it's unclear, what this effect represent. It just represents a difference in the mice and the healer brain during baseline versus treatment. You
There's
know,
some intention to connect. I mean, may I ask you just one quick follow-up question, which is the sham healer trying to connect with the mouse and trying to give them healing? Or what is the sham practitioner doing?
now the sham practitioner is just holding his hand, like the healer. They're just resting. They're not doing anything.
Right. But they're focusing in on the mouse and they're
They're
putting their
focusing on
hands
the
on the
mice.
page and everything, right? There's a shift in perception, a shift in focus, a shift in
Yes,
intention
yes.
towards some kind of connection. So when they're connecting, regardless of whether it's a trained healer or not a trained healer, and regardless of whether the mouse has cancer or not, when there is this focused connection, you see a synchronization between the brains of human and animal.
Yeah, we see a synchronization in the brain. What you said is one interpretation. Because we also see that in the sham, it's hard to say, oh, this is the healing intention or not the healing intention. It's just like basically during the treatment period, you have some change in the human brain, you have some change in the mice brain, and there is some commodulation. There's some synchronization. And so it's been observed in human as well. Human watching each other.
Yeah.
If I see you, our brain are going to synchronize. So maybe the fact that both the healer and the sham are focusing on the mice, and I wouldn't say necessarily making eye contact, but there is like the body stance that the mice can feel. So
Yeah,
that
I mean, it seems to build on those hyperscanning studies of people who just look at, what happens when there's some sort of shared dialogue, shared connection, when singing together, dancing together, you see increased synchronization in those situations as well, between humans at least, right?
yeah.
Um,
And again, the last one here, that's the most interesting column because that's the interaction. Is there something special about the biofield therapist treating the cancer mice during the treatment period? And it tells you, yes, there is between these pair of electrodes. And of course, we are doing many statistical tests because we have many pairs. So we have to do the right correction. And we do. We use the special tools to correct for the fact that we're doing many, repeated many statistics. And this is the difference. So we see in the baseline, we see the effect is bigger in both cancer and control, but still, it's larger for cancer than control. We see this dots is bigger right here, and it's smaller during the treatment, but this is significant. These are not significant here. Um, so that's interesting in itself. Same for these ones here at higher frequency. So just to conclude, so we have a little bit time for question. I don't know if we need to end on the hour or if we can have a few more minutes. Uh, but these subtle effects, because these are subtle effects, it's like what I didn't mention is like to get enough statistical power. We asked so it's all science is all about repetition. We asked the heater to come back 60 times. 60 times we wire, so we don't wire the EEG 60 times because they do 4 sessions at a time. It takes the entire day.
mhm.
So we aim for statistical power to be able to deduce things on which we can say, oh, this is robust statistics. But so these are subtle effects because it comes through a lot of repetition. You don't see them in the raw data. But I believe this is just the tip of the iceberg. Like I like this analogy. This is a vase from ancient Greece where you have electric eel, basically, and they knew about electricity. They use it for healing, but it was just a curiosity at that point. And I think we're at the same stage. For us, it's just a curiosity, but it has huge repercussion for the study of consciousness. Um, and yeah, thank you for listening to me. Thank you for the BR Foundation and Emerald Gate who supported some of these projects. Uh,
Amazing. Wow, amazing, Arnaud. Thank you so much. Like a big virtual clap and applause for I'm just listening to you. So grateful that you decided to move your inquiry into consciousness through science, and what a gift it is to the Biofield community that you have chosen to dedicate your efforts in this sphere, because really it takes some phenomenal scientists to be doing such careful, rigorous, and such exciting work. So exciting. And I agree. Tip of the iceberg. Tip of the iceberg. So for those of you who are listening, before we get into questions, who are thinking, how do we scale up this research? Please come talk to me. Come talk to Arnaud. We can give you many ideas of different organizations, including the Consciousness and Healing Initiative, Institute of Neuronic Sciences, and UC San Diego, of ways that we can continue to carry this work forward. There are many scientists that we know of personally who would love to come and help replicate this, and many healing practitioners as well, as we start exploring other healers, other traditions, other approaches. This is really opening the door to an incredible program of research. And what I find so incredible about it is one of the values of the biofield in this time in our study of it is to help awaken humanity to the potential of our shared connection, and how deep that really goes. What a critical thing to be doing at this time. And thank you, Arno. I have a few minutes if you do. We won't get to all 20 of your questions, y'all, and I apologize. Maybe we can have Arno back again. Hi, Cheryl. Great to see you here on the chat. So lovely for you to join us. We have some fellow incredible biofield scientists with us as well. Really quick procedural question that always gets asked, and the first one here, is it possible to have a recording? Yes. If you joined this and you registered for it, you will get a replay link, and you are free to send this. Please do send this to others. The replay is absolutely free for the entire weekend. It is our gift to you as the nonprofit Consciousness and Healing Initiative. Please share this widely with your colleagues who are wondering if there's any real data and where the cutting edge is of biofield research today. Share it with your healing practitioners, share it with fellow scientists, share it with people who are decision makers in the hospital for them to understand the real science behind this. And for those of you who would like to support these kinds of dialogues into the future and the present, please consider joining us as a contributor. When you do, you will have access to this recording and about 70 plus more with leading scientists, leading healing practitioners, inspiring folks with practical tips as well as scientific insights. Jason will put that link in the chat. You just go to chi.is, join/join. Tax deductible donation of a mere 108 a year. That's $9 a month. Better than a green juice even. And so we cannot thank you enough. It is because of contributors, and thank you to all the contributors who come and continue to support Chi. It is with your support that we are able to keep this flowing. So thank you so much for considering that. I hope you will join us as a contributor today and support the science and education of healing. Now, to some more questions. Here's a question from Katie. What do you find are the biggest barriers in developing, conducting, and publishing research studies on this topics? Is it IRB, ethics, repeatable methods, subjectivity of experience? I would add funding. What would you say, Arnaud, are the biggest barriers at this point to doing more of this type of research?
I think the main barrier is funding.
It is.
And
Mhm.
ethical approval is not a big problem. Yeah, I mean, as they look more at the study design, whether you're going to, do something that's rigorous or not, than, necessarily the exact questions you're asking. It's ethical committee, it's not going to judge necessarily the science. Yeah, funding is critical. This study, cost, it came 60 times. You need to have staff, and grow the cells and maintain the mice. And so yeah, it's just expensive to run these studies.
Yeah, so when we want rigorous studies, we have to be able to pay for them, and that includes staff, it includes the materials. We have to be able to do that. Sometimes people are like, wow, studies cost a lot. Well, they do to do them well. And it's great to start doing piloting in our own labs, in our own basements, and just sort of explore ideas. But then when we want to take it to this level of, publication in nature journals, everything has to be so tightly controlled, so rigorous. that it does take a lot of, as we can see, dedication and hard work and
And
resource.
yeah,
Yeah,
and resource. And the plots I showed you with it looks easy, like the human brain and the mice brain. This is I programmed everything from scratch. It's like there's no software to process mice and human brain. So all of this is yeah, programming and and statistical software to get that out, the lines, etc. So that's why it's still consuming also. It's like there's, you have to use sometimes tools which you have to design yourself. That's the process of science, and that's why we love it. But it's also, you can't just do that like, oh, I'm just going to spend 1 hour. We also often have, potential donors. It's like, oh yeah, it's just easy, but no, yeah, it takes a lot of time because everything is kind of custom. You're asking questions. This is questions never been asked before. You have to create the method. You have to create everything from scratch.
yeah, yes. And how lucky are we that you have that skill and capability to lend to the field and choose to put it in this direction. So, so grateful that you can do that. And look, we know scientists, just like healers, it's a labor of love. We love doing this stuff. So it's not a burden for us to do it. We just need to eat, we do it. So, okay, here's a question from Sharon, and I think we want to clarify this. And she may have gotten the answer to this, but I just do want to clarify because she said, why not analyze the cells, not just the healer? But yes, he did analyze the cells, and maybe Arno, you just want to emphasize what were the cell findings in your study? What actually happened to the cells themselves?
So yeah, in the paper, I don't know if you shared a link to the paper.
You did.
Here's
Yeah.
a figure on the what happens to the cell themselves. And to tell the truth, I'm not really competent. I'm the signal processing brainwave person. I'm not really competent. I think the figure did show some changes. When the heater was. Uh, so this is
What
it's
I remember about this, and my memory might be incorrect, but what I remember about this is that there was a significant shift in the calcium flux of the cells between the control cells and the live cells, so that the calcium flux actually decreased, if
yeah.
correct me if I'm wrong, Arno, in the cancer cells. And that's important because we have more and more data now suggesting that there's almost a over communication between electrical communication between the cells. So a decrease in the calcium flux for the cancer cells treated by the healer would suggest more of a return to homeostasis. You could say, in a way, kind of better boundaries if you wanted to kind of talk about it like that. I may be wrong, but I think that's what I had gotten from that paper.
Yeah, so that's the main figure in the paper. Biofield first therapy markedly inhibited the invasiveness of pancreatic cancer cells. And so we can see treated here and control. And this was repeated twice. And the reason I didn't show it is, I don't feel competent. Like if there's a question like right now, it's like,
Right, 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 right. Well, and
what are the details? I don't know how,
well, that's the beauty of these transdisciplinary teams. We had a psychoneuroimmunologist who is Lorenza Cohen, with people who have expertise in cancer and people who have expertise in electrophysiology, which was the focus of your talk today, all coming together for these studies. I mean, this is how we make science. I mean, this is how we evolve science. This is a very intriguing and excellent question by Ellen, and we will stop in a minute with these questions. We want to honor your time, everyone's time. Mary Ellen asks a very simple question that I will answer. Can Reiki be considered a type of biofield therapy? Absolutely. Look, biofield is a term that we Western scientists decided to start using to cover therapies, including Reiki, healing touch, pranic healing, Jo-Ray. All of these in the science world are considered biofield therapies. We stopped using the term energy because energy drops off at a distance, and we felt like that maybe wasn't the right term to be using for these healing therapies. But yes, Reiki is considered a biofield therapy. Okay, last question for today by Ellen and Arno. Maybe we'll have the pleasure of having you back sometime, which would be amazing because there are a lot of questions here. Um, do you think that the biofield is the cause or the effect of the reflection of the exchange of energies between the non-material energies and the manifested material, meaning the bodies, I guess, or something else? And by the way, if that wasn't enough, and how might this relate to the observer effect in the double slit experiment?
Yeah, that's
What a question.
and my answer is usually I'm not a theorist. I let off the people, so I'll get the results, and then there's when you publish a paper, there's many different ways to interpret the results based on different models, and yeah, so I don't know how to answer that specific question. I mean, I have my own model of how consciousness interact with the world, but yeah, that's very high level, and that probably wouldn't answer that question.
Yeah, more time than, but really a great question, Ellen, for consideration, maybe something to take through our day. What is informing the field? How are we co-informing the field? It's a really brilliant piece to ponder. I have three suggestions for all of us today with this brilliant presentation from Arnaud. Arnaud, thank you so much. We're all allies of the field. We're all interested in the field. We're coming to it from curiosity, from experience, many different places with our connection with the biofield today. There are three things that you can do today and this weekend to support the field. One is to champion this work. And championing that means, standing in your own authority about your relationship to the field and how you work with it, whether it's in business, whether it's in healing, whether it's in science, whatever. The other way is to invite people to learn more. Sharing this webinar is a brilliant way for you to do that, or sharing your own work. These are ways that you can invite people more broadly into the field so that they can help co-inform the field of healing. And I would like to invite you to invest today. Whether you choose to invest in the science or the education, we hope you will join us as a contributor if you're not already. Again, chi.is/join. You will receive monthly meditations in your inbox. You'll receive the latest science updates and all access to all of these brilliant presentations as our thank you for supporting the science and practice of healing today. And please look into our dear friends at the Institute of Noetic Sciences if you are not familiar with them already, and consider supporting their work as well. Incredible science team there leading incredible work. So, Arnaud, thank you so much for your time today. Always such a pleasure, and I look forward to seeing you soon at UCSD as well. And many blessings for continued incredible research and impact for the field. Thank you.
Thank you. Yeah, and one last word about what you said is we had a collaborator at university, and one of his students says, "Oh yeah, I'm interested in ions and what they do, etc." And he was like, discarding, "Oh no, it's not really. This is pseudoscience, etc." And then he brought this professor the paper. Look, this is what they published in this mainstream journal. And the professor completely changed his stance. He's like, my gosh, this is incredible. And it's just, this is the way some, not all, but some scientists work, data, it's like data, but you let the data speak basically. So if
Mm.
you have friends which are scientists, and they are like dismissive, just, send them the paper and says, what do you think of that? And, have them, go deep into it with ChatGPT or something and find flaws.
ChatGPT, Notebook LM, and this webinar, right? Hear from the horse's mouth. Hear the perspective, hear the rigor. Thank you all so much. Thank you. You're getting a lot of love in the chat, Arnaud. Thank you for spending time with us today. And you all have a beautiful weekend. We will see you soon. Tune in for another beautiful webinar next month with Dr. Mark Mincola. Thank you, Arnaud.
Thank you.